Hello, good morning, viewers at home. My name remains Olatunde Michael, and this is another opportunity for us to um, come for another lecture series on this Omega Bio-Edutainment. Don't forget that we have actually started this long ago, about two or three previous meetings. This is talking about the fundamentality of microbiology. That is, what are those things that are very, very essential that you should know, and how and what should be our school of thought, what should be our belief, what are those things that we actually need to know as a microbiologist about the origin of life. Don't forget that everything biology do wherever, anytime, any day, we are the only science that is the science of life. We deal with life, we protect life, we talk about life. And no matter how small that organism could be or that life could be, microbiologists, we are poised to make it, guide it, protect it, and study it as well. Don't forget that in the past I have told you that we talk about the WOESI classification. That is, all living organisms, how can they be classified? And which I told you that based on the ribosomal RNA classification, which is so many authors have attempted a lot of classification, and before they will be able to classify organism, this time around, it doesn't actually mean that it has to be microorganism, any life at all. If you are going to classify them, you must base them on certain characteristics. For example, I might want to classify the students in biological sciences. The first thing is to look for what are the similarities. So that similarity will now make you to be able to place them in the same group, and those differences will now make you to be able to place them in different groups. For example, we might just want to say that biosa is the collective name for all biological students in faculty of science, and that is what combine them together is they are studying biology. Is it going to be microbiology? Are you talking of environmental biology? So biology is a common denominator that make them to be classified together into the same group. And the one that will not differentiate them will now be in terms of you will now divide them into environmental biology and microbiology. So that is exactly the principle in biology, or which is called the science of naming, which is taxonomy. We are going to see all this as we actually progress in the course of this study. Don't forget that this course is microbial ecology and evolution, and this is the continuation of the lecture 2 in this course. And when we talk of one of the most controversial topic in the whole world, in all fields till today, is origin of life. How does life originate? How did it actually originate? So we there are a lot of school of thought about that. That is talking about evolution in an attempt to study the origin, starting from the origin down to what you have here is studying of evolution. And don't forget that previously I have said it in discourse that evolution has to be defined as development with advancement. You develop along the line. This was not the way, for example, now where the whole world is actually evolving from is that we are evolving from analog age, and we are developing, we are advancing towards the digital age. So that will actually give you what evolution is. And ecology, which has to do with the interrelationship. Microorganisms exist in communities. Are you talking of viruses? Are you talking of bacteria? Are you talking of fungi? Are you talking of parasites? Are you talking of protozoa? Whatever organism you are actually talking about, they must exist as a community. So, and if they are existing as a community, that is to tell you that evolution, that is to tell you that ecology is the study of the interrelationship. Okay. Now, previously in this course, we have been able to talk about understanding the scope and importance of microbial world. When we come across microbial world, you should be able to understand it. Then you should be able to also have a chemical understanding of microorganisms. That is, if you pick up a microorganism, no matter how small they could be, you should be able to look at that microorganism from the chemical perspective of it. And that is the reason why all microorganisms, or all organisms, so to say, they are actually have certain major components: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and so to say. So if you gather all the organism, be it microorganism, they cannot survive with any of this. When you synchronize, that is, when you break down their cellular components, when you break them down into fractions, what you will see is that you will see that they are made up of basic chemical principles. Then also we are able to look at prokaryotic versus eukaryotic, which we have actually talked about. Then we also talk about metabolism, studying of metabolism, growth and control of microorganisms. That is, by the time, if you are able to understand the way microorganisms respire, if you understand the metabolism, if you understand the catabolic processes and anabolic processes that take place in microorganisms, you will be able to know how can that be affected when it is come to how do they grow and how can you actually control them. So what we are saying in essence is, and I've said it in the first lecture, that lecture one, that in this course, we are going to look into the details of studying microbiology, or studying this course in terms of their evolution and ecology, so as to be able to help us in order to see how it can be controlled. Then apart from that, I also said that this course is going to culminate into a simple principle of recombinant DNA technology, which is also termed as genetic engineering. Recombinant DNA technology is that. So, without wasting our time, we look at why microbiology matters in Nigeria and also all over the world. Apart from that, we also look at a certain basic definition like microbiome, microbe, and pathogen. I say wherever you talk about patho, anything patho is talking about diseases. Gene, that is the gene, that is gene or organism or microorganism that are capable of causing infection or disease in a certain host is called a pathogen. And microbiome means the collection of microorganisms and their genes in a certain habitat. That is, if you are trying to study the soil, all the collection of all microorganisms there, and if you are studying them and you are collecting them with respect to their genes, that is to give you the collective word is known as a microbiome. And symbiosis, we look at that, commensals and opportunistic. I told you that opportunistic, from the word opportunist, that means they will be they are harmless in a natural or in their normal state. They do not cause any infection, they do not cause any discomfort. What they do is to assist in relationship, coexistence with you in order to be able to protect you. But once your immune system is low, once your immune system is dampened, so they will take that as an opportunity to become pathogenic. That is, they will take that to be an opportunity to harm you. And that is the reason why what we are saying, for example, Mycobacterium tuberculosis is an opportunistic infection in immunodepressed individuals. Ordinarily, when you inhale Mycobacterium tuberculosis, under normal circumstance, your immune system, macrophages, they will just go and hit them up, or they will become so latent, that is, they will not cause any discomfort. They will be hiding somewhere, and they can be there throughout your lifetime. They will just exist with you without causing any infection, without causing any problem for you, and that is what is called latent TB. But immediately the immune system is low, immediately there's an opportunity for you not feeding well, immediately there's an opportunity that your immune system is depressed, so they will take that opportunity, and microorganisms that have coexisted with you for a very long time without causing any infection will now cause infection, start causing this. So these are opportunistic relationship. So we also look at key milestones and a short story about that. We look at a pure culture, which was introduced by Coach Postleat. and which is still very relevant up to today. So we also look at a microbial diversity. Wherever you talk about diversity, diversity means differences, differences that they coexist together. Microbial diversity can be viewed from the angle that it can be differentiated into bacteria, archaea. When you differentiate them into bacteria and archaea, these are prokaryotic without membrane-bounded organelle. And eukaryotic microbes, these include our yeast, our mold, and protists, which consist of algae and protozoa. And when we are looking at acellular agents like viruses, virions, and prions, all these, they are absent. They call them this. Don't forget that today, we now have more the modern day classification. I've told you at the beginning of this lecture that its microorganism can be classified. Let me say generally, classification of a life or organism can be classified into three. The first one is archaea group. The second one is called the bacteria, and the third one is called eukarya. Eukarya. We are going to see more of this when we go on. And I told you earlier on that you have to understand this lecture or microorganism from the perspective of a chemical, that is atoms coming together bounded by certain bond, which would be van der Waals forces, covalent forces, ionic forces, hydrogen bonds, and so on and so forth. And you still have to know that irrespective of how small microorganism could be, water is still very special to them in terms of their polarity, in terms of the hydrogen bonding, in terms of them being unifier solvents, and the cohesion forces that you have around them. So we also look at it's very, very important for microorganisms to exist with these certain ranges of pH, where you introduce buffers in order to be able to stabilize or equilibrate alkalinity and acidity. We are going to see more of this when we go on. You should know that microorganisms are made up of macromolecules. And what are these macromolecules? These include carbohydrates, lipids, protein, and nucleic acid, which could be, which are RNA and DNA. Then enzymes. Enzymes are very, very special. Enzymes are proteinous in nature, and they have optimal condition in which they will be able to perform well. So biological catalysts, this speed up the rate of chemical reaction or biological reaction without being consumed. They will take part in that reaction, and they themselves, they will not being consumed along the way. So, and we will also need to remember, just a kind of recall what is meant by active site, specificity, cofactors and coenzymes, and temperature and pH. That all these are factors that affect enzyme performance. So we actually look at a prokaryotic cell, a typical prokaryotic cell, the architectural plan of a prokaryotic cell. We are able to look at it, and we look at it from the angle that starting from the way their cell wall is, or the component of their cell wall, they can be classified into gram positive, gram negative, and into acid fast. When you see gram positive, the difference between gram positive and gram negative is that gram negative have very thin peptidoglycan plus their outer membrane. And when we are talking about gram positive, they have very thick peptidoglycan, okay, and also contain with absence of outer membrane. And waxy, that is mycolic acid or acid fast, they have a special addition to their peptidoglycan, and that is mycolic acid. And that's why the cell wall has to be disrupted by the use of certain acid like hydrochloric acid, and that's why they call them acid fast, because it is that acid that will first of all be used to dislodge, to disrupt, or to destroy the mycolic acid. So it is the hydrochloric acid that is now reacting with mycolic acid that will now be able to allow certain dyes to be able to pass through. But don't forget that based on their cell wall structure, bacteria can be, or prokaryotic cell can be classified into three. That is gram positive, gram negative, and acid fast, which is the presence of mycolic acid. And typically, we can see that this is a prokaryotic cell which contains inclusion in form of endospore. Some of them have endospore, that is, they form endospore. And you should also know that a ribosome, they have 70 S. Don't forget that we don't have to waste our time because on this. And the same thing I've said all over is what this slide is telling us. Teichoic acid, that is, and you can see the peptidoglycan layer. You can see teichoic acid on it, but you can see that this one does not have a teichoic acid, but it has a very thin. When you compare the thickness of this one with this one, you will see that the thickness from here to this place, that is the layer, peptidoglycan layer, and it is the same layer here that you have. Okay, you can see it here. Very, very thin. Very, very thin. So, but this one has a lipid. This one has a lipid um, phospho layer here. Then you see the acid, which is a membrane. Okay. Then prokaryotic cell like a gram-positive, gram-negative, acid fast, and peptidoglycan. You can see this is what makes them to be different from each other. And what I've said so far is the internal structure of a prokaryotic cell contain cytoplasmic membrane, which is used or useful for transportation of energy. I mean, for transportation and energy generation. And nucleoid, which is the circular chromosome, which consists of which it can be linking to the chromosome we have in human. Although don't forget that prokaryotic cell, I told you that they do not have a membrane-bounded organelle. They are not specific. So, but they have nucleoid, which consists of a circular chromosome or circular DNA. And there are some microorganisms like bacteria prokaryotic cell that, in addition to the circular chromosome or the circular DNA, they have what is called plasmid. And what are plasmid? These are extra chromosomal extra DNA that they have in addition to the main DNA. This is the main DNA, or what we can call the main chromosome. But certain bacteria, certain prokaryotic, they have an additional component that is called plasmid. And any microorganism, any bacteria that has a plasmid, you should know what they are using that plasmid to do mainly is for them to survive the harsh condition of antibiotic. So they are mainly used for antibiotic resistance. So, and what this one is telling you is that if you have two microorganisms or two prokaryotic cells, one is having plasmid and the other one is not having. The one that is having plasmid is going to resist more antibiotics than the one that does not have it. You should know that. Then the ribosome in prokaryotic cell, the ribosome in prokaryotic cell is 70 S. 70 S. S stand for subvert. So we are going to see this later on. So this is the anatomy of what I have been saying so far. So prokaryotic cell can be divided into the envelope. The first thing is the envelope. The first thing you will when you are coming to the okay, it's just like when you are coming to the whole building. Imagine prokaryotic cell like a whole building. Okay, so the gate that you are going to see in order to be able to enter into the house, this is called the envelope. The envelope, and that envelope contain peptidoglycan layer. Some contain outer membrane, and some of them, like mycobacteria I told you, they contain mycolic acid. And the presence of this mycolic acid, we know the implication, we know what they are using it for. So the envelope, the internal structures or the internal features, which include the plasmid I've told you, the inclusion body, the endospores, the ribosomes. Then the surface structures like the flagella. Okay, this is an example of a flagella. The fimbriae, that is a cilia, fimbriae, just like is cilia like. Then you have another layer which is called the glycocalyx. Glycocalyx, you should know Like glyco is telling you that it is from glucose. So we can see this as we go on. So this is a typical cell surface. We have seen it. The fimbriae or pili, it is used for attachment during sexual or what is called a conjugation process. And the glycocalyx is a capsule, slime capsule or what is called slime. So, and this is what they use in order to be able to attach firmly into the to their host. So we have seen this. So membrane bounded. The difference between eukaryotic cell and prokaryotic cell is that this one contains membrane bounded organelle like nucleus, endoplasmic reticulum, and Golgi apparatus. And eukaryotic also has mitochondria and chloroplast. We will see more of this when we go on in this course, but we are going to see how relevant is mitochondria and chloroplast. But you should know that mitochondria and chloroplast, they are two uh, brothers and sisters. One is found in animal cell, or and the other one is limited to plant-like. It might not be animal, plant-like. That is it. And we will also in this course, another thing that's very, very important is endosymbiosis. Endo, we will see it as we go on there. Instead of a 70S ribosome that we have in prokaryotic cell, this one has 80S ribosomes, and you also have a cilia and flagella. We will see this, and this is the anatomical structure of a eukaryotic cell. You can see that this one, they have very definite shape. You can see the Golgi apparatus, you can see the mitochondria, you can see the nucleus, you can see the flagella, you can see ribosome, you can see chloroplast, you can see cytochondria, you can see microtubules, you can see microfilament, you can see intermediate filament, and so on and so forth. So this one contain membrane bounded organelle. You should know that. Okay, so we don't have time to actually go for that because it's what we have seen. So this is the architectural plan of eukaryotic membrane. We said eukaryotic cell wall, their whole, instead of being made with a peptidoglycan, the cell wall of fungi is made up of a chitin, and that in algae is either cellulose or silica, and the other one is a protozoa, which is made up of a pellicle. Okay, so we have been able to differentiate between prokaryotic cell and eukaryotic cell. And why? What do they actually use each of these structure for? That in the genome of a prokaryotic is that it is circular and they don't have nucleus. But in that of eukaryotic, instead of their DNA or their to be linear, to be circular, this one is linear, and nucleus is very, 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 very important in eukaryotic cell. It's always present in eukaryotic cell. And what is it being used for? It is only being used for gene regulation and replication. So we have passed this. We will see as we go on. We also have passed through this. So we were able to also look at the big picture of metabolism, that it is either it is can be classified into breaking down and building up. That is catabolic and metabolic anabolic processes. And we also see that ATP still remain the energy currency of the cell, be it prokaryotic, for all processes. ATP is the energy currency. Then when we talk of other secondary currency that you can also have, apart from ATP, is you can have it in form of NADH plus and so on, FAD and so on. And we also look at what glycolysis is all about, and glucose to two molecules of pyruvate plus a small amount of ATP and NADH. We have seen that glycolysis is the conversion of glucose via 10 enzymatic processes from glucose to 2 pyruvate acid. And I told you, or maybe I mentioned it, that there is always a puzzle, and the puzzle is, why is it that pyruvate is the central or is the key for metabolism, for cellular respiration? So we will see this. I said and I expected that you must have been taught, or you are going to be taught in microbial biochemistry. You will see a lot of this cycle. You will see how glucose can be converted into CO2 and water. You see fermentation, you see the differences between fermentation and anaerobic processes. Okay, we see the essence of acetyl CoA, and we look at the differences between Krebs cycle, carboxylic acid cycle, as well as glycolysis. So, just like I said, fermentation versus anaerobic process. So, we see that in fermentation, there is always regeneration of NAD plus, and the end product is usually lactate, ethanol, and or other acid. But in anaerobic, that one uses non-oxygen acceptors like nitrates and sulfates. And the application of these two is in food industries, diagnostics, and ecology. Okay, so we were able to also differentiate between phototrophic and chemolithotrophic. Okay, then we also examine microbial growth. I told you that the lag phase is the phase at which the microorganism is getting used. Don't forget that microorganism is being introduced from the natural environment, like sample. If you are collecting sample, soil sample, that is the natural environment of that microorganism. And when you are now you are introducing it into the lab, that is, you want to culture it, it is going to, that means you must be able to provide an enabling environment for that microorganism to be able to grow. Not only grow, they should be able to grow well. Just like just imagine that you want to rear or you want to domesticate a lion, or you are taking any animal that you need to make your pet. You are taking it from the jungle, so, and you want to let it live with you in your house. That means the first time you are going to bring that animal into your house, it is going to be very, very, very inactive. So sometimes it can even affect his feeding pattern. It can even affect to the level that sometimes it can even get emaciated. So lag is very, very similar to this stage. That is the stage of acclimatization. That is the stage of learning. That microorganism is just learning about the new environment. It's just acquiring a kind of a additional DNA to be able to help them to adapt into their new environment. That is the lag phase, and that is why growth at this level is non-appreciable. It is not. It is not notice. It is not unnoticeable at this level. Okay, then it's going immediately after it has acclimatized. Immediately after it has learned a lot about its new environment, it will now begin to explore. It begins to radiate. It begins to shine. If the color is white, it will bring out that color to actually tell you that it is enjoying the new environment. And that is the time that it will now remember to produce the more. It will now remember to regenerate itself. And that is the log phase. Log phase. That is when there is exponential increase in sizes and number. So, and that is the brilliant time that microorganism will be able to perform well. And that is the log phase. So, the log phase will now lead them to the stationary phase, the stage at which there will be no more regeneration, no more development, and that is when the environment, the metabolic waste of the microorganism will begin or might begin to be very, very toxic to them. So no growth will be noticed. So it's going to be on a straight line, and it's going to be at that peak. That is stationary stage. So after the stationary stage, the next thing is the death phase or the death stage. So at this is when there will be drastic reduction in the viability, drastic reduction in number of certain microorganisms. So, and we know that that is exactly the principle behind drug production. So when you so it The pattern the pattern of this curve will depend on when the drug is being introduced. If the drug is introduced to microorganism at the exponential stage, you will just discover that you might spend you might take more than the dosage before you be able to. So but when it is entering at the lag phase, okay, when it is entering at lag phase, you just see that this lag phase will just kill everything, so it won't be able to proceed any further. It won't be able to proceed any further, so it will just enter from lag phase. Then the next one for effective drug, from lag it will just enter the death stage straight. So that is the effectivity or the effectiveness of any therapy. So that any immediately at any point in time, if your drug is being introduced at lag phase, log phase, or stationary phase, that means if it is introduced from here, the next thing is that it's going to enter into the death stage phase. Oh, that means your antibiotic is very, very, very effective. Then, if from log, that is at that point you introduce it, that means there won't be any stage like stationary. So from log, it will just enter the death stage. So that's why we said the microbial growth or how microorganisms actually develop or grow can actually give us a clue, certain clue onto how we might be very successful in order to be able to control that microorganism. Don't forget that we are in the class of microbial ecology and evolution. And don't forget that I have repeatedly told you that evolution is development with advancement. And I said ecology is the interrelationship, the study of the interrelationship of the microorganism. How do they interact with their environment? Do they interact in form of symbiotic association? Do they exist in form of parasitic association, or are they commensal? So all these are very, very important before we will be able to have a very good clue about what we are talking about. And I have told you, and which we have said, that the pattern of growth of microorganism would depend on whether our therapy or our control strategy is effective or it is not. Okay. Now we also talk about growth requirement. Growth requirement before you will be able to grow well. Before people will see you and they will see that you are actually feeding well. The same thing with microorganisms. There are certain requirements, including the oxygen. So I told us that in the last time that the first one is a temperature. Sacrifice, this one exists at low temperature, and the mesophile middle, like the room temperature, and thermophile they require thermo from the word high heat. So that means they require high temperature for them to be able to grow. Do you know that there are certain microorganisms that naturally they will grow in a very hot environment? No matter how hot it could be, they will grow at that environment. These are called the thermophiles. Then pH and osmolarity, that is talking about the salinity or the content of salt, of salt, yeah, that is being required by certain microorganisms by way. And based on their oxygen requirement, they can be classified into obligate aerobes or obligate anaerobes. When you say something is obligate, that is, it is a must that they will exist as oxygen lovers. It is a must that they will exist as non-oxygen lovers. So, and when we talk about facultative means they may be found here and they may be found there. They are in the midway. That is, they can live as obligate aerobes or obligate anaerobes, and they can also live as a facultative. So they can exist, they can tolerate certain amount of oxygen, and certain amount of oxygen can also kill them. So that is that. Then oxygen, oh, sorry, the next one is a micro aerophile. Micro from very small quantity, aerophile, that is oxygen. That is, these are microorganisms, or these are organisms that tolerate very small quantity of oxygen, and that's why we call them micro aerophiles. Then we also have aerotolerance. Tolerance, they tolerate in the absence, in the presence of oxygen, they will still exist. They tolerate it, but naturally, that is not what they are. Okay. So media types, we talk about this very well, and we are still going to talk about this. So we have it can be in form of, that is, when you are taking microorganisms into the lab and you want to study them, how will you culture them? What food will you prepare for them? Media are food that microorganism eats. So, and for example, whatever food you are, as human being, whatever food we are going to eat, will contain certain requirements. Is it going to be of the class of carbohydrates? Is it going to be mixture? Is it going to be balanced? So that is exactly what is called media. And whenever you come across, I've told you, whenever you come across anything aseptic, aseptic means, or aseptic is a condition that remove contamination. Aseptic, removal of contamination. So I want you to know it from that angle. Then we will also get used in our practical. We will also get use of a pure culture and a stick plate method as we go on. So all this is what we are going to know about. Then measuring. How will you measure the growth of microorganism? It's not that you have to go and use a tape rule. Yes, certain time you will use something like a measuring tape to be able to measure them. So you can measure them in form of viable count. Viable means they are still living. They exhibit all the characteristics of a living organism. That is, they'll be able to reproduce, they'll be able to respire, they'll be able to remove their waste, they will be able to they will be able to perform all the characteristics of living things, and that is when it is said that they are viable. Somebody who is viable is only the person that can eat. It's only the person that can sense the environment. It's the only person that can communicate with the environment. So the same thing with microorganisms. So they can be measured on their viability. We call it viability counting, and it is measured in CFU. In CFU, C is for colony, F is forming, colony forming, U is for unit. That is, the viability of a microorganism will depend on the number of colonies that they are forming. So that means if you have one colony today, if it is still going to be viable, still going to be regarded as living, that means before you come tomorrow, you must have seen more than one colony that will be added. So that is what is called viable count. Then another one is serial dilution. Serial dilution, the essence of serial dilution is to be able to serially thin down the number of microorganisms. So is to be serially reduce the number or the population of microorganisms. So you will always start from one, that is minus one. The first you, if what you do, what is a serial dilution, is you have your sample already in form of a liquid. After that, you now arrange like 10 test tubes. You arrange them serially. In that one, you must have put 9 ml, measure 9 ml into each of these tubes. It could be 9 ml of water, it could be 9 ml of any other substance that will be able to support the growth of that microorganism. So you just pick, to the first one, you pick 1 ml of that sample, you introduce it into the first one, that is onto minus one. So when you add 1 ml plus 9 ml content or liquid that is already there, that's going to make number one to be 10. So after that, after you must have gently aspirated or aspirated it, so the next thing is to remove that 1 ml again, put it into the second flask. In that, it's going to be minus two. So that of second flask is now also going to be 10 mil. Out of that 10 mil, you take another 1 mil, you introduce it to the third flask. That is going to be -3 at that point in time. You also add it to make the third test tube to be 10 mil. So you also reduce, you will also remove 1 mil from it. You introduce it to the fourth, fifth, sixth, and seventh, to the last flask. And the more, as you are moving from -1 to this, is the you are tilling them down. The population will be reducing gradually, and that is what it is called serial dilution. For example, now you want to, okay, let's say you have a liquid, milk. You know the color of milk. Let's say you now have milk, and then you have now serially uh, arranged your test tube. So you want to dilute them. You want to dilute the milk. So you remove 1 ml of that milk into one. You will see that one, the color that you have in one will be very close to the original milk that you have there. And sometimes as you are moving up like that, you are moving from um -1 to 10, you just discover that the milky color or the milky coloration will be disappearing. When you get to the 10th one, it is going to be as if you are dealing with ordinary water, so which is going to be colorless. So that is what is called serial dilution. So which is it? Then we also have, there are two types of plate preparation. We will also see this in our practical. Don't forget that practical is going to start very, very soon. Maybe next week or upper week will begin. It's still going to be, there are some we do online, and there are some of these we do it online. We give you instruction online, then you carry it out and you will submit it when you get to this. Okay, and it's very, very important. Okay, control of microorganisms. You should be able to know the differences between sterilization, disinfection, and antisepticis. Okay, when you are talking, if you are talking of sterilization is that you are removing contaminants from an environment. You are sterilizing it. For example, under normal circumstance, sterilization process is when you begin to mop, you clean your table before you are sorry, that is a disinfection. Sterilization means that for example, when you are removing microorganism from your equipment, you sterilize it by the use of autoclave, by the use of high temperature or high pressure, to be able to remove contaminant, to be able to remove microorganism right away from there. And when you talk of disinfection and antiseptics, the difference between disinfectant and antiseptic substance or liquid is that disinfection, you are using it on inanimate objects, inanimate like tabletop, like Isaiah. You cannot use that on somebody's skin. So, and antiseptics are those ones that you can use for on the body. So that is that. Then the use of heat, that is autoclaving, high heat pasteurization. So we will see more of this when we go. And don't forget that you must have even been taught or been used to this when you are doing STM 210. Okay, 201, that is a general microbiology. So although we will see all this when we go on, then chemical agent like alcohol, hydrogen, we will also see this in, you will also discuss more of this in the pharmaceutical microbiology when we go on. So we will be able to know quartz is an acronym for four different chemical agents, which we are going to see that. Then the recombinant DNA is such that you is the manipulation of DNA in order to get your desired product. And that is you should be familiar with the structure of DNA at this level, the central dogma theory. You should be able to go and refresh your understanding about it. The central dogma theory is when you have the DNA, this will be converted into RNA, transcribed, transcription through the process of transcription. You have messenger RNA. So from that messenger RNA, it will now go into amino acid. So the movement from DNA down to amino acid, it's what is called the central dogma theory. We will see all this, and you will also be taught of that in microbial metabolism or microbial biochemistry as well. So restriction enzyme, we will have to get used to all this when we go to DNA structure of this. So by now, I will not I will just introduce you to recombinant DNA technology until we are able to study long on this. We'll come back to this, just like I said, for this. So, and these are step by step when you want to manipulate the DNA. That is, you isolate the gene, you cut the gene, I mean DNA vector with restriction enzyme. And I told you we are coming back to this. So that time, I'm going to really bring you a kind of images that is going to help you to be able to understand it more. And PCR, don't forget, I also mentioned this PCR technique in general instrumentation. PCR is a polymerase chain reaction. I told you that polymerase, that is an enzyme that polymerizes, enzyme that uh, expand or amplify section of DNA, and it can be divided into four different processes. The first one is denaturation. Denaturation, that is, you will increase the DNA, you will increase it to high temperature. High temperature is going to help you to increase it. Then after that, annealing means you are going to allow them to join together. Don't forget that the DNA is a double-stranded molecule. So when you are denaturing it, that means the two strands that have been together like this, you are separating them. That is the process of denaturation. So when you have now unsettled them, when you have denatured them, when you have removed, when you have converted them from being joined together, when they are now single-stranded, so they will be looking for a complementary strand. They will be looking for sequences that will be able to make them just like somebody that has been a couple, let me say, that has been in marriage before, and they now have a kind of divorce. uh Divorce. So those one, when they want to look for husband again, they will be so aggressive that somebody who has remained to be single. So the same way, when they are together, denaturation means you are separating them. And when they have been separated as a result of high increase in temperature, so the next one is that the temperature will now be brought down so as to make them to be able to do what? Look for their own partner. So this one will look for his own partner or complementary basis. The other one will also look for his complementary basis. Then this process will now continue. Process that started with a single stranded DNA. Now, when it has separated, this one will look for his partner, another one will look for his partner, and it will be double like that until you have two raised to power 32. That is, if you have 32 cycle, 32 cycle. So after annealing, allowing them to join together to look for their partner. So the next one is to extend them. So then this one can be done in a thermal cycler, so which they call the PCR machine. So we will get used to all this. I think we should be able to learn this when we get to in general instrumentation. That is when we'll be talking about polymerase chain reaction and electrophoresis. So electrophoresis separates DNA according to their sizes. So those ones that have very high weights, they won't be able to move far. They won't be able to move far. And the one that are lightweighted, they will be the one that will move far. We will see all this by the time we go on. Okay. Now, what is the application of bio mole- um, so what is the implication of recombinant DNA technology or cloning? So it can be used in biopharma, that is to be able to diagnose, I mean to be able to treat people that are diabetics. We also use it in the diagnosis in vaccine, then very, very applicable in environmental biotech and so on. Then in bioethics, where you will see the consent of your subject and so on and so forth. And GMO, that is genetically modified labeling. We will see this the more, and you come across all this. Don't forget that this is still your elementary experience or elementary stages in microbiology. So, and that's why you are going to get used to all this by the time you also go on. Don't forget that I told you that this course is microbial evolution and ecology. So now you know the difference between prokaryotic and eukaryotic. You know the principle behind gram staining, classify them into gram positive, gram negative, and acid fast. And now you also know the basic principle of genetically modified or genetically what is called genetic engineering, and what we can call the recombinant DNA technology. You are aware of this. Then we also look at growth. At what point can you make use of growth? At what point, what are the oxygen and other requirements that is necessary for microbial growth? We have seen that. We have actually seen that your curve will actually depend on the presence or absence of very tolerable environment. So that then now the origin of life, you should be able to know very well that okay, what I can still see because of I don't want you to I don't want to take our time. I can still see that we still have one more lecture, that is one more hour lecture or thereabout to be able to go, to be able to finish it, because this is just the 37th slide, and we have like a 50 something slides that we are going through. So sincerely, I want to stop here today and stop to be able to continue from where we are stopping. And by the time we come back, we will just go straight to origin of life. I'll just review very briefly, and we go to how life actually originated in the aspects of a microbial or microbial world. Okay, so we will be able to look at what do we mean, the hypothesis, the RNA world, how does it actually come about, then what can we learn from hydrothermal vent model. So, in explaining the origin of life, how can we make use of RNA world to explain the origin of life, and how can we also make use of hydrothermal vent to be able to explain this? But then again, chemical evolution with the belief that all complex chemicals of today must have evolved. Don't forget that I told you about evolution. So they believe that most of the carbohydrates, most of the particles that we have today, that if you pick up all any substance, any molecule, if you pick it up today, and you just discover that they are made up of carbon, hydrogen, oxygen, and all this one. So they believe that there was a time that there was nothing like carbohydrates. They believe there was a time nothing like nucleic acid. They believe that there was a time when there was nothing like lipid. There was a time that there was nothing like organelle. So they believe. So we are going to look at some of this school of thoughts, and we'll be able to use it in order to explain the origin of life. And by the time we use that to explain the origin of life, you will now know where you want to belong. I and how do you want to actually know about microbial world. So we will see all this in terms of this. Then also we will also look at the origin of life from the conditions, from condition around us, reducing atmosphere. There are some people that believe that there was a time that our atmosphere was not like this, was not like this. That the some people still believe that this sky that you are looking at, there was a time that it was not as tall as this. So we will believe in, we will look at all these erroneous school of thought. And don't forget that if we study microbiology without talking about microbial evolution, without talking about where are they coming from, how do they look like that time, that means even if we want to study them in their present environment, we will not be able to know where they are coming from. So there was a time that they existed in form of just reducing atmosphere. They believe that okay, so we are going to see all this when we also go on. Then we will look at what do we mean by classical species stroke microbial species. What do we mean by strains? So what we call classical species in taxonomy, not in microorganism, I mean microbiology. So we are going to compare all this and be able to move forward from there. So don't forget that I told you we'll talk about endosymbiosis, oxygenation of the heart, and so on and so forth. So without wasting our time, don't forget what you should do in the comment section. Please be of very great time. I want to wish you all the best in your and have a great weekend ahead. Don't forget to talk about comments in the comment section. Thank you and God bless you.